So everyone is having this string input file. Did you prepare this in your system?
Yes, ma'am.
Yes, ma'am.
Okay. Sanya, Sonali, what about you?
Yes, ma'am.
Yes,
Okay.
ma'am, we have it.
Okay, fine. So we can start with the network analysis. So any one of you, like, do you know anything about this network? So in
Ma'am,
this, mm-hmm.
during micro array, ma'am, we did network analysis.
Okay, so do you like you have some idea about this?
Yeah, ma'am.
Okay, it's fine. So all of you like have done this network analysis.
No, ma'am, we
No,
have.
ma'am.
Okay, let's see. Then what you can do, just copy this gene name and go to the string database. Just open that. Now you can see in the Excel file you have like multiple genes, right? So here in this string you cannot only perform the gene or proteins, you can also perform with the gene also. So like you are having what the multiple genes, right? So you have to select this, then paste the gene names here and just select the organism, then search. Now you can see like in your Excel like total hit was 90, right? So again you can see the 90 query items you have put here. Okay, you don't need to do anything here, just continue, then it will make the network in the string database, right? So now can you tell me guys what is this circular shape and what are these lines?
The lines show that they interact with each other.
Okay. Oh, Arya, what is
Uh,
this?
the circle are the nodes and the lines are the edges.
Okay, so you can see here the network has been generated and we are able to see some circular shape, right? So that we called it as the node, and inside that you can see in the circular shape or the node you can see the structures, right? In that, so we should be able to understand what those structures are and why these nodes are highlighted with the different colors, correct? So if these two nodes are connected with each other by some lines, so these lines we call it as the edges. So with the help of the edges, we can see the interaction between the two genes. Okay. That means what the gene is controlling the expression of the other genes. Okay. So all those information, if you want to read, you can just select this legend option and you can see this. What is this node color here? First you can see the colored node. So colored node will represent the different categories or the properties of the protein. So why it has coded? Because in order to so that we can identify or analyze specific group of protein with the network, that is why they have colored the nodes here. And again, you can see the query protein and the first shell of interactor. What is this first shell of interactors or the query protein? Anything? You know? So query protein means this is the protein of the primary interest. So it might be colored. And the first shell of interactor means what? These are the protein that will directly interact with your query protein. Whereas in the white node, it is referring to the second shell of interactor, which is typically representing what? The protein that are two step away from the query protein. So second shell interactor means what? The protein that is interacting with the first shell of interactor but do not directly. It is indirectly connected with that query proteins, not the directly way. Whereas when you see the empty node, so empty node means it is representing the protein of unknown three-dimensional structure, which is signifying that the arrangement of the atom in that protein has not yet been experimentally determined. So there is an absence of structural information for a particular protein. That is why it is an empty node here. Next is the filled node. Filled node is used to represent the protein with the known or predicted 3D structure, which is indicating the presence of structural data for a particular protein. That is why, like in the field node, you can see this structure. So, in the structure, it is having what? The 3D dimensional structure of that protein. Okay. Next, when you see the edges, like in this color, which is indicating the what? The from curated databases, which is basically referring to protein interaction data that has been carefully selected, organized, and validated. Next is the experimentally determined protein, which has been directly observed or measured through laboratory experiment that you can see with this edges color. Next, what is this gene neighborhood? Can anyone tell me what is this gene neighborhood?
Genes located nearby each other.
Okay. So yeah, so in the gene neighborhood, what happened? Genes are located close to each other and more likely to be involved in the related biological process. So that is what the gene neighborhood. And when you will see the edges with the red color, that will indicate the gene fusion. Gene fusion is what? When the two or more genes are like fused together, combining together, and which is resulting what in the hybrid gene, which is having what the novel characteristics or the properties. So that is what the gene fusion. Next is the gene co-occurrence. So gene co-occurrence means what? uh, Whenever like genes are appearing together in the same genome and they are sharing the similar gene expression pattern, that is what the gene co-occurrence. So whenever next you will see the yellow edges, that is what the text mining. So for that particular genes, you will get some articles and the research papers. Next is the co-expression. Co-expression means what? Two or more gene are expressed at similar level under the specific condition that will be indicating co-expression. Next is the protein homology. So protein that are homologous, often they have what? The similar structure and maybe the function, and also they have the common ancestor. That is what the protein homology. Clear? Anything doubt in this gene neighborhood text mining? Is this term is clear? Done? Okay. So whenever like we'll select this network, it will appear this network. Okay. Again, if you will select this experiment, in this section, can you see the medium word here, exploratory? Can anyone tell what is this color is indicating, medium and this? So in this experiment section, it is giving you what the protein interaction. So here in this first, you can see the protein interaction was detected using a tandem affinity purification assay. This is one of the biochemical process which is basically used to isolate and identify the protein that interact with the specific protein of interest. Now you can see here the average detection confidence is exploratory. So whenever you see this word, it will indicate the interaction may require further validation and investigation to confirm its significance and reliability. Okay. Next, if you see the medium word here, so that interaction has been observed, but it is reasonably reliable, but still it requires further validation for its significance. Clear? What is the difference between this medium and the exploratory? Here, like it is just giving you the gene names here and only the assays like from which they have been isolated the protein. So that assay they are giving you here. Clear about this experiment section?
Yes, ma'am.
Okay. Next, databases. Yeah. So again, like in the databases, you can see the annotated pathways, right? So annotated pathway in the KEG are what the graphical representation of the interconnected steps that are involved in the specific biological process. So in this database, like it is just giving you the database name here, KEG, Reactome. So these are like particular databases from where they have taken the gene information. So all those pathways, or you can say the database, are listed here in this. Okay. And Reactome is nothing, it is just a database is again, and it is one of the curated knowledge base of the human biological pathway. Okay. And it is the name of the pathway again, it is giving you below. Next, in the text mining. Text mining is what we have discussed already. Like when the edges are like in the yellow color, it will indicate the text mining, right? So here in the text mining, you can see a list of significant protein here, right? This protein names or maybe the gene names, right? So it has been extracted from the abstract of the scientific literature. So here you can see the abstract here, right? So here you can see in the text mining, the title and the abstract of the publication are displayed together with the link to the publication. So you can see, like you can, if you want to open that, it will directly like navigate to the PubMed. So you can, if you want to read that, you can read that in the, for this. Next is the neighborhood. Yeah. So anything you know, like what these figures are indicating, if it is taught this database.
No, ma'am, I don't know.
Okay, okay. So in this gene neighborhood, you can see the genes which are like located in a run, which is linked with the black color line. Can you see here? This one. Can you see my cursor?
Yes, ma'am.
Okay.
Yes, ma'am.
So here you can see these genes are linked with the black line, right? And if there are other gene in the run that are below, you can see here, it is indicated with the white triangles here, right? So basically, these runs are below the certain score threshold. So here in the gene neighborhood, if I select this, you can see It is giving you the gene name 3970. Remember this. Now, next to that, you can see 3971. Can you see this gene neighborhood?
Yes, ma'am.
So, gene neighborhood definition is what? Genes are located close to each other, right? So, you can see before it was 3970 and next to that 3971. Can we say these are gene neighborhood here, right?
Yes, ma'am.
Yeah. So, next in these triangles, if you select this, so 13816. So, can you see the IDs are varying, right? Correct. So why it is varying here and why it is giving you the white triangles here? Because they are not that much closely related to each other. Okay. So if you are getting only single black line, so that are you, like you can say these are gene neighborhood. Okay. So these are somewhat gene neighborhood, these particular lines. Clear? And if you want to read more information, you just select that gene. You will just navigate to those information. Clear about this gene neighborhood?
Then the direction about.
See, don't see the direction. Here. It is not linked with the direction. You have to see whenever you see the white triangles here, that means that particular genes are certain below the current score threshold. That is why like it is giving you the white triangles. Okay.
Okay, ma'am. Okay, ma'am.
Next, just select the fusion here. So all the genes are highlighted with the different colors. So if you want to see, you just need to select that particular information. If you want to navigate, just select that. It will just open that. Okay. And you can read more information regarding that gene. Now in this gene fusion, you can see the species in which fusion is occurring are listed towards left. You can see all the species name here, right? Sometime what happened, like here in this data you can see all the this figures are highlighted with different colors, right? Sometime you will get the white gene also, okay, white colored here. So here it is not white color, so white genes will indicate what? The fusion is not directly with each other. Okay, so that's it all about this fusion. In the co-occurrence, you can see the presence of the protein in a species. Species are listed here. And you can see the red color square here, right? And the absence with the white spaces here. So whenever you see the red color, it will reflect what? The proteins here. And if the intensity of the red color in a square is more, that will indicate the amount of conservation of the homologous protein in the species. That is what the red color square here. If you are seeing here like white square here, that means there is a absence of protein, okay, in that particular species. So you can see this is a particular gene here, right? You can see if you go down, so in none of these species it is present here. Can you see that? Right? But if you go down, like it is little bit present in this species, correct? But here if you see for this particular gene, again, like till here none of the gene it is present in this species, but it is more here, right? And if it is red, darker red, that is giving you the amount of conservation of the homologous protein. But in this figure, can you see the black color also? Right? What is that black color now? Do you have any idea?
No, ma'am.
Okay. So this color black is often like used in the gene co-occurrence only. And this black color also reflect the high level of interaction between the two genes. Okay. So actually, like if we compare to the red color, black color is more like giving you what the co-occurrence. It is having like more relationship or the conservation, you can say, in the black square. Okay, comparatively to the red one. Okay, clear?
Yeah, ma'am. Got it.
Done. Is it clear about all this section? Legend. Any doubt here? Again, in this settings, you don't need to do anything. You just put the default. And in the analysis, like it is just giving you the scores, how many nodes and edges are giving you. Like you don't need to worry about that. Now, next thing is like if you are reading the research article, like do you find this type of network in that research article? No, we are finding that figures in the proper format, right? We cannot put these type of networks in the research papers. Okay, and also in the string database, like there is one drawback. For example, if I want to highlight my upregulated and downregulated genes, that things I cannot do in the string databases, right? So for that, we are using the another database, or you can say another tool. Okay, tool you can say. That is what the cytoscape. So in the cytoscape, you can edit according to your interest. Okay, and you can give the colors also there, and you can highlight the upregulated and the downregulated gene. Okay, so this is one of the drawback in the string. So for that, now you have to download this file, which is opened in the Excel and the Cytoscape. Can you just download this in your system? Is it clear, like all about the string database? Any doubt here?
No, ma'am. Understood.
Okay, fine. Just download it and let me know.
Done, ma'am.
Okay. Don't download the latest version of this cytoscape, okay? So shall we start?
Yes, ma'am.
Okay. So first thing is we need to import the file, right? So just go to the import, then network from file. Yeah. Now can you see the network here in the cytoscape? All of you? You can try like later on. Just look at my screen. So now you can see the network here. So in this string data, like cytoscape, what we are going to do like we are going to highlight our upregulated and the downregulated genes. Okay, so here you can see all of the genes are like highlighted with the blue color here, right? So with this, we cannot say anything like is this gene is upregulated or the downregulated, right? Again, in this network is what it is very compact. You can see in this figure the gene is overlapping, right? We can move also like this, right? So now we cannot see the gene name properly here in this figure, correct? Now I want to expand the network. So for that, you have to go to the view option. Then there is a option show tool panel. Now I don't want to select any one of this. Just uncheck this and drag it. Can you see it? The name of the genes now?
Ma'am, did you open the same file that you downloaded or is this different?
I will tell you. I will tell you. You have to perform like later on. Actually, this file which I have taken from the same data. But in your case, like in your file, the 50 lakhs reads are I've cut, right? But in this is my previous data which has been taken from same title only, okay? But I have like more than like 90 genes here, okay, in this file. In order
Okay,
to
ma'am, got
just
it.
to yeah, I just need to explain like this type of data you are getting in the cytoscape when you are having lot of significant genes, okay? Clear? But
Yeah, ma'am, got
you guys have to open like later on when we'll discuss the entire session on this cytoscape. We will try on our real data, okay? Just see the option like which you can follow in this cytoscape, okay? Just look at this. So now like you can expand also, right? But the data which we were looking, so now I will import my real data, okay? The data which you guys are having, okay? But in real time you are getting that complex network only in the cytoscape. correct? This is the data like you are getting, right?
Yes, ma'am.
Okay,
Yes, ma'am.
fine. Fine. Now, if I want to give the like entire pathway or entire network the yellow color, so that you can do with the help of style. So there is a default mapping and bypass. First, we will see the default option. So by default, it will just give the entire color to your network. So for example, I want to fill the color of my network with the yellow. Make it okay. Now you see it has been changed to yellow, right? Again, for this network, if you want to change the shape, so that also you can do. Make it to rectangle. And you can see properly it is in the rectangle shape, correct? Clear? Any doubt you can tell me.
No, ma'am.
Okay, so do you know about the hub genes? What is the hub gene?
Ma'am, the gene which has most number of edges.
Okay, so now I want to highlight the hub genes, okay, in this network. So hub genes are those which is having the maximum interaction. So how we will know which genes are hub gene? So for that purpose, we should know the number of edges, right? From the number of edges, we will come to know the higher interaction, right? Correct. So in the node table, like we can see only the gene name here. Can you see? Only the gene names you are able to see. But if we want to get the edges information, so for that you have to go to the tools here. Then analyze network. Make it okay. Now, if I scroll towards this left, can you see the degree here? Just double press here. And can you see now I have make to the descending order. You can see the 4 number. 4 number is indicating what the interactions or the number of edges. Now, if I want to highlight my hub genes, just copy that particular gene and then paste that. Then enter. Now, if I want to highlight this, because now I cannot see, right, that hub gene. So now you can see the bypass option is opened, right? So for that, you can fill the color and make it to pink maybe or the purple, anything. And just click on the panel here and you can see the hub genes here. Can you see? And also for this particular genes, what was the number of interaction? 4. And here you can see the 4 interaction. It is interacting with the 4 genes. Can you see that? Again, like if you want to highlight your this gene, which is having what? The third, like 3 interaction. Again, just search it. It has been selected. Bypass is open now. Again, you can fill the color. You can see here. Clear about this hub gene? What are those hub genes? Now, I want to see which gene is controlling the expression of other genes, right? So now I want to highlight my source also and the target, right? So now can you tell me what is the source gene? What is the source gene?
Mm,
So source gene is nothing, it is just controlling the expression of the target gene. Okay, so that I'm going to highlight now. Okay, source and the target. So that you have to do with the edge. So you can see here in the below, there is a edge option. Okay. Now for the source, if you want to give any shape, okay. So for example, I want to give this delta shape, just apply that. Now you can see the source. Now if you want to change the shape of the target, you can just select that and make it circle. Just apply that. Source and the target will be indicating with this different shapes. Got it? Clear about this default and the bypass? Bypass, it will only open when you are selecting any particular gene here. Okay, then only you can just highlight that. Otherwise, you just have to use the default option only. Clear? Anything you
Yes,
can
ma'am.
apply. Okay, clear till here? Um, Pushpit, Sonali, Nuparna, Sanya, what
Yes,
about
ma'am.
you guys? Okay. So now till here what we talk about, like how to change the color, right? How to change the shape, source and the target, and what are the hub genes we talked, right? Now the important thing is to highlight my upregulated and the downregulated genes, correct? Now you can open your input, this string input file. So here you can see in this file, like together you are having the upregulated and the downregulated, correct? So for this you have to apply what, the filter. So for the upregulated genes, the filter was what? For the log2 greater than or equal to 1. Make it 1 here. Okay. Just copy this and paste in another sheet. Just rename it as the upregulated genes. Now again, go back and put the filter again for the downregulated genes. It should be less than or equal to -1. So now these are my upregulated genes. Okay. So now what you have to do, you just have to copy the genes from here and go to cytoscape and put that in the search box. Then enter. You can see the selected nine nodes here and the bypass option is open now. Now I want to give the red color to my upregulated genes. So what you can do, just go to the node here and fill color and make it red. Okay. Now you can see it is not red, right? You have to click on this panel, then only it will highlight. Can you see my upregulated genes here?
Yes, ma'am.
So can you see the difference between the default and the bypass? In the default, what it is doing, it is by default, it is applying color to all the genes, right? But whenever we are just giving the gene name here, the bypass is open, then you can fill the color to the selected genes, correct? Clear? The difference between the default and the bypass here. Now, same thing you have to do for the downregulated also. Just copy the genes Again, paste it, enter, node, fill color. So now you can see the green color. So these are my up-regulated and the down-regulated. So this is how you are highlighting the genes in the cytoscape. Clear? Any doubt till here?
No, ma'am.
Okay. Now it's up to you. You want to change the shape, transparency, width. Like there are a lot of options you can apply to your mm, this network. Even like in the layout, you can see the different layout they are giving you. If you want the grid, you can use that. If you want this type, like it's up to you which type of layout you need. You can see that. Okay. So in real time, it is not in this format. Okay. In your system also. So whenever like you want to publish the article, we are like putting in the organic layout. So here you can see this is the Y files organic layout. So this format we are using whenever we want to publish the paper. Okay. Or whenever you want to put this picture. So like so in the some of the system, like you are not having this organic layout, right? You need to install that. Can you see in your system like it will be written install Y files? So if somebody is not having this Y files organic layout, you need to install that. So I will tell you how to do that. Just go to the apps option. There is a app manager, and in this search box you have to just type Y files. And in my system it is already installed. So you need to select that and you need to install that in your system. Can you? Done till here? Did you install this Y files in your system?
Ma'am, I already had it.
Ma'am, I have installed it.
Okay. Okay. What about Yash, Pushpit?
Done, ma'am.
Do you have guys? Sonali
Ma'am,
ma'am?
I have it. Yeah, I have it.
Okay, fine, fine. So now you need to select this layout, Y files organic layout. Just select that. You can see this is my proper format of the network. Now I need to export also from this cytoscape, correct? So for that you have to go to the file section here, and there is an option export. Then what you can do, there is an option network to image. Go to that. Just zoom it to 500, because in the cytoscape whenever you are downloading the image it is not disturbing your pixel of the picture. So that is why you can zoom it to 500%. Make it okay. And then you can see fine. Any doubt till here?
No, no, ma'am.
Okay. Now I will tell you about this mapping part. Now what you have to do, you just have to import file. Wait a second. So just go to this file section. Then there is a option import, and import there is option table from file. Now what I did, I have just saved my this string input file in this CSV format, because in the cytoscape it is not taking this workbook file. Okay, so that is why I've already saved that in the CSV. You can do it later on, just look at in my system first, then just open this in your system. Okay, so I've just loaded this. Now you can see the mapping one. It is open, right? So now what we are going to do, with the help of this log to fold chains, we are going to highlight our genes. Okay, because currently we are just, what we did, like we just pasted the gene names, right? And we just gave what the colors, right? What if I want to change the palette of the color here to the network? So that time just go to this mapping here, and there is an option column. Just select the log2. On the basis of log2, I'm going to change this. Then mapping type. In the mapping type, you have to go with the continuous mapping here. And you have to provide what? The cutoff. Now, minimum is what? -1. And maximum is 1. Make it okay. Sorry. This is the... Yeah. In the fill color, okay? You have to go not with the height. Give the range here. Then you can select the palette from here. So it's up to you which color you want to give to your network. You can just select that. For example, I just want this one. Make it okay. Yes. Now you can see, like if it is more red color, so that is what my upregulated genes. If it is in the yellow color, it is somewhat like it is not showing any expression here, and down regulated is more the green color. Okay, and you can see. Clear how to do the changes with the log to fold change here? Any doubt?
Ma'am, for this, we need to first convert our Excel file into CSV, right?
Yeah, same file, array you have to do. You have this file, right? This one, right?
Yeah, ma'am, I have this.
So you just need to save as just browse it.
Ma'am, I
And
got this part.
yeah,
But when I'm doing this, so we have like three sheets, no, in that one, like one for upregulated and downregulated. So it's showing that if you're having multiple sheets, then it can't convert to CSV.
so you have to give this one a cutoff. So you haven't this one Excel sheet, this one. You don't have to put this upregulated and downregulated genes, because already like you have the cutoff there in that file, right, along with the genes. So here in
Okay,
the
got it.
cytoscape you are putting the cutoff. Now you don't need to put the upregulated and the downregulated genes in the separate, otherwise it won't accept. Should I share this file, CSV file, with you?
No, ma'am, I got it.
Ma'am, can you show once more how you did that log to part?
Yes, ma'am. So, ma'am, here you can see in the default option, there was a option fill color, right?
Yes.
So I just selected this fill color option, and in that you can see the column here. Then in the column, there are a lot of options they have provided, right? But I want to change my network according to my log to fold change, because I'm providing the genes along with the log to fold here, right? So just select that log to fold change, and in the mapping type there is a continuous mapping. Just go with that, and now you can see the graph here, right, the color shade. Just double click on that. Then there is a set minimum and maximum range you can put according to your cutoff, okay, that you have provided for your upregulated and downregulated. Then if you want to change the color, go to current palette, just select this and make it okay. Yes, okay. Now can you see? If it is towards the more this blue color, that means what? These are upregulated genes, right? More. And if it is pink, then it is what? The downregulated. Clear?
Yeah, yeah.
Yeah. And same thing, like if I download directly this, it won't download in the entire network. What you have to do, just zoom in. If the entire network is there, now it is perfect. Now then you have to export, okay? Then only it will give you the entire network. And the same way, like you have to export network to image. Okay, sure. Done. So any doubt, guys, here in this string and the cytoscape?
Ma'am, I'm getting the color as blue and orange. So how did you change
Uh,
that?
you can share your screen, Arya.
Yeah, ma'am, I'll do it.
Hmm. Wait a second, just cancel it. Did you set your cutoff? Go to that set minimum and maximum range, Arya. No, click that. Yeah, below that there is option. Yeah, you need to write here -1 and 1, because you have provided -1 for the down regulated. Yeah, 1. Make it okay. Correct. Now you just choose the palette. Yeah, any palette you can just select. Make it okay. Mm, yes. Make it okay. Yeah. So now in this cytoscape, you can see if it is more towards the orange. Is it orange or red? Like I cannot see properly. Is if it is more towards orange, then it is your upregulated genes according to your log to field change. And if it is more green color, it is downregulated genes. Okay, clear?
Yes, ma'am, got it.
Okay. Another gene which are highlighted with the different colors, no, maybe yellow color. So they are not the significant genes here. Like you can say they are not like in the criteria because you have provided the cutoff there. Any doubt? You can practice the cytoscape and the maybe the string. Like if you have any doubt, we are left with one session only. We will meet to like maybe on Monday now. Then it's all about this RNA-seq. Okay, if you have any doubt, we'll discuss on Monday.
Okay, ma'am.
Okay.
So, ma'am, tomorrow we'll be having the test on RNA-seq.
Uh, Maybe yeah, like you guys will be having. That HR will tell you guys. I don't have any idea.
Okay, ma'am.
But they will inform you like on the group. Okay. So yeah, we can meet on Monday and have a great weekend, guys. Enjoy. Take care.
Thank you, ma'am.
Okay. Bye bye.
Thank you.
Yeah.